Okay, great. Well, thank you all for coming this morning, and thanks Tim and Rick for the invitation to talk. Hopefully what I'll try to get across to you this morning is some of the future and where we are going with MALDI imaging, and I'll use applications in ocular tissues to try to get those points across. So what I'm going to talk about, I don't really need to spend any time on the methodology because it's been covered already. Fortunately, it'll save me a little bit of time. Then I'm going to talk about lens proteins, an area we've been working in for quite a long time. I'm going to talk about soluble proteins, the crystallines, and integral membrane protein, which is not so easy to image. And then I'll move on to optic nerve imaging of lipids, and then some metabolites in AMD, which you just had an introduction to by Tim. And lastly, I'll talk about this bottleneck in terms of identifying what it is, what analytes are we actually imaging. So I don't want to spend too much time on the methodology other than to say we're using frozen tissues. In most cases, except for the flat mounts of retina, we're doing frozen sectioning. We apply the matrix either by spray coating, high density droplet array with a portrait spotter, or also with sublimation for some of the lipid imaging. And then, of course, ultimately we end up with our MALDI mass spec images. I'm not going to talk today about manual spotting of matrix and just profiling to get protein profiles. That's for a different talk. So, as I mentioned, most of the tissues that we work with are frozen, and we take frozen sections, 10 to 20 micron thick sections, and the washes are very important. As you've heard multiple times this morning, this keypad, there we go. So the sample preparation is absolutely essential for making this work and getting information that's useful. So the washes are very, very key for membrane proteins, as you'll see. And then in answer to Steve's question of just a minute ago, we use ammonium formate or ammonium acetate washes, which coalesces the lipid signals down to mostly the protonated form and increases our signal intensity for any given lipid species. And I just mentioned how we apply the matrix. So I'm going to talk about 3 ocular tissues. So I'm going to talk about the lens in the anterior chamber of the eye, and then I'm going to talk about the optic nerve in the back, and then also some retina imaging. So those are the 3 tissues that we're going to cover, all very, very different in their nature. So in starting with the lens is a very, very interesting tissue. It's pretty simple. It contains 2 cell types. It's a single layer of epithelial cells on the anterior surface, right here. And then these epithelial cells differentiate and elongate into what are called fiber cells. And these fiber cells that are in the center of your lens are as old as you are. So there's no protein turnover. And the fiber cells that are newly differentiated on the periphery of the lens are contain relatively newly synthesized proteins. So you can think of it sort of as rings on a tree. We have proteins that are young on the outside, ranging in a gradient to as old as you are in the very center of the lens. So the question we're trying to ask here is, how do the proteins age? What changes occur as we age? And then ultimately, how do these changes cause a cataract or an opacification of the lens? So the first example I'm going to show you is for lens alpha A crystalline. It's a soluble protein, about 20 kilodaltons in molecular weight. The crystallines are extremely abundant in the lens. They're about 300 mg per mil, so very, very easy to see in our MALDI mass spectrometer. And if you look at intact alpha A crystalline, it has 173 amino acids. In a 7-year-old lens section, you can see that it's predominantly in the periphery of the lens, or the lens fiber cells, in the young fiber cells, and there is some alpha A crystalline present in the center of the lens. But we also can see some truncation products. So we see a 101 residue truncation product in the center of a 7-year-old lens. We see 58 residue product and a 40 residue product, both in the center of the lens. So this pattern will continue throughout the aging of the lens. So if you look at a 29-year-old lens, you can see that there's less intact alpha A crystalline in the center. The signal is lower intensity here compared to a 7-year-old. And the 1 to 101 product is mainly in the intercortical region, and it degrades further to these smaller truncated products. And there are many other truncated products which I'm not showing you here, but are filling up the center of the lens. So then, by the time we're 51 years old, there's very little soluble intact alpha-A crystalline in the bulk of our lens. And you can see the 1:101 product is also sort of a ring-shaped distribution, and it's degraded into these smaller products in the center of the lens. And then, and then if you go to even older lenses, you see the same, and even the small products tend to degrade further. You can start to see this ring-like formation here. So the center of the lens in older humans is filled with these small crystalline products. And other investigators have shown that some of these small peptides are pro-aggregatory, and some are mini chaperones that can prevent aggregation. So it's very, very important to know where these products are forming and what the products are to understand the biology that's going on. So in a very, very recent study, we looked at age progression of different small peptides, presumably crystalline products. And so we go from right to left here, 7 years, 37-year-old, 78-year-old, and then a cataract lens. And the goal of this study was to look at age increases in peptide products, but also to see if we could see anything that was unique to cataract. So before I get to that, so here we see a 2100 Dalton peptide in the 78-year-old human lens, not very much in the 70-year-old cataract, but it's quite abundant in the 78-year-old lens. We also found cataract-specific peptides. So at 2892 and 3542, there's an abundance of these peptides in the cataractus region. These are nuclear cataracts in this 70-year-old lens. So then the next obvious question is, what are these peptides? What is it that we're imaging? And I got an email last night suggesting that the data has been acquired but has yet to be interpreted. So to be determined. Hot off the press. So the other protein that we're very interested in the lens is an integral membrane protein known as aquaporin 0. It's a very, very abundant protein in the lens. It's a water channel, and you can see these alpha helical regions here span the membrane, the plasma membrane of the fiber cells. And so our lab has been working on this for a very, very long time. We've identified most of the major modifications, and what I'm going to show you today are some images of this major truncation product at residue 246. So as we age, the truncation at this particular site increases. And we also were able to identify lipid modifications to this lysine and the N-terminal methionine. And I'll show you the localization of where those modifications occur. So imaging, or actually getting signals from an integral membrane protein by mass spectrometry, is not easy. So our approach to imaging this integral membrane protein is to wash the highly abundant soluble crystallines away. So we take our lens section, put it on a MALDI target plate, wash extensively with water, and now with 0.1% formic acid, and then we spot the matrix, you can see here, with a robotic spotter in high density, about 200 microns depending on the size of the lens, and we use 90% acetonitrile as our matrix solvent. And if we apply this approach, this solvent matrix combination without washing, you get a very ugly MALDI mass spectrum. There's lots of crystallines present and a little bit of signal for aquaporin 0. And the resulting image is not very informative. But if we wash the tissue first, wash the crystallines away, and then apply the matrix in this 90% acetonitrile, you see a very, very nice signal for aquaporin 0. It's about 28.2 kilodaltons, 263 amino acids. There are still a few crystallines present. We can't get rid of them that easily, but nevertheless, with this type of signal, we can generate an image. And so that's what I'm showing here. This is a signal for intact aquaporin 0 in an 11-year-old human lens, and it's highly abundant in the young fiber cells, and it very, very quickly degrades where this red line appears. So the center has some aquaporin 0, some intact aquaporin 0, but it's less abundant than in the very young outer shell. And the center of the lens in this 11-year-old is filled with this 1 to 246 truncation product. So, and I can tell you that if you look in older lenses, this center region is completely devoid of intact aquaporin 0, and it's filled up entirely with these truncated forms of the protein. So using imaging mass spectrometry, we're able to determine what the truncation products are of the major lens proteins and exactly where in the course of human lens aging these processes occur. So I mentioned the fatty acid modification to the aquaporin 0. So we've found this both in bovine lenses and in human lenses. So this is the unmodified aquaporin 0 in a cow lens, and I showed you this image earlier for a human lens. And here's where the fatty acid modification occurs. It doesn't occur in the very younger fiber cells. It occurs after a certain period of time, and then it gets abundantly modified. So roughly 50% of the protein is modified with a few of the different lipids. And the same is seen in the human lenses. So this is the distribution or localization of where the fatty acid modification occurs. And it doesn't actually go away in the center. This is due to the fact that the protein's truncated. So the truncated forms are also contain the fatty acid modification. Okay. So moving on to the second part of the talk, looking at optic nerve. This is a collaboration with Dr. David Calkins in ophthalmology at Vanderbilt, and he's interested in glaucoma, which is the second leading cause of blindness worldwide, only second to cataract, and it's first among African Americans. And so a standard hypothesis in the field is that elevated intraocular pressure ultimately, over long periods of time, leads to damage to the retina ganglion cell layer and to the optic nerves, eventually leading to blindness. So we were interested in imaging both the retina and the optic nerve to see what changes we could observe using MALDI imaging. So Dave Calkins and others have generated a number of different mouse models for glaucoma, and in this case we're looking at a mouse optic nerve section. So you can see here, this is the optic nerve. This is a tissue section now coated with DHB matrix, dihydroxybenzoic acid, so it's a little bit hazy, but this is where the optic nerve is, and then you can see here is the retina right here in a mouse. And so I'm showing here an image of a lipid species at 760.6 mass to charge units, and this is, again, sublimated with DHB. And this is a positive ion mode, and if you look at just the mass spectrum that you can get from this tissue, it's very, very rich in information. There's a lot of very, very strong signals for lipids, and we can image any one of these signals and then identify them based on the techniques that Tim just told you about. So in negative ion mode, you get some of the negatively charged lipids, and here is a slightly different section. These images, I should have said, are taken at 10 micron spatial resolution. So here's the optic nerve right here. Again, the retinal layer, and this particular species is located in the periphery of the optic nerve tissue, and it turns out through accurate mass measurement and tandem mass spectrometry on an FTICR instrument, we can get fragmentation of this lipid directly out of the tissue. Turns out that this is a sulfatide species, so a negative ion has got the sulfate group here on the lipid head group. So the sulfatides are enriched in the myelin sheath, and that's what's coating the outside of the optic nerve. Now we can also take cross-sections through the optic nerve. It's actually easier to take cross-sections than try to get a longitudinal section straight through the optic nerve, because these things are very, very small in diameter, about 200 microns, as you can see here from this scale bar. So here's an image of a lipid that fills the center of the optic nerve tissue. And we identified this, again, based on tandem mass spectrometry as a phosphatidylcholine, and it turns out that the ultimate structure is PC18-1-18-0, as shown here, present in the bulk of the optic nerve. Oops. And then here's another lipid distribution that shows, again, a peripheral distribution, and we were interested in this image because there's another signal here for this lipid, and this turns out to be an adjacent blood vessel. So this is a lipid that's present in a blood vessel that's adjacent to the optic nerve and runs parallel to the optic nerve. So you can start to tease out different anatomical features at this spatial resolution. And here's the structure of this particular signal at 811.0. So we are also interested in layers of the retina and where the damage occurs there. So we've developed a method to take cross sections of a mouse retina, and at 10 micron spatial resolution, we can tease out, using lipid imaging, 8 of the 9 cell layers present in the retina. And so here's the ganglion cell layer in orange, so we should be able to see changes in the lipid in the glaucoma models. And also of interest is this single cell layer, the retinal pigment epithelium shown here in red, that is very important in the next topic that I'll be talking about. So at this spatial resolution and with this capability, we should be able to look in the mouse models and find changes. So the last topic I'm going to talk about is age-related macular degeneration, and Tim already talked about this briefly in his presentation. There are 2 forms of the disease in humans, the wet form and the dry form. As Tim mentioned, this is the leading cause of blindness in older Americans. And it's believed to be due to the accumulation of retinoid metabolites that are present in the center of the eye, in the macular region. But there's no current method for imaging specific retinoids. The standard approach is to use fluorescent imaging and look at this, the formation of fluorescent material. But as you know, the fluorescence that you get from a particular tissue is not due to any single species. It's a combination of different fluorophores. So in collaboration with Rosalie Crouch and Joel Oblonzi at the Medical University of South Carolina, we developed a way to image these retinoid species. So the buildup of lipofuscin, which is the fluorescent material, a complex mixture of molecules, occurs in the retinal pigment epithelial cell layer. So here's the back of the retina. These are the photoreceptors here. And so it's this area that we're interested in imaging. So just like Tim, we take flat mounts of the retina. We peel off the neural retina layer, that is the ganglion cells and the photoreceptors, exposing the RPE layer to the matrix that we then deposit. And then we image where the species are in the retina. So the main retinoid metabolite we're interested is called A2E. It's so named because it contains 2 molecules of vitamin E, vitamin A, and then 1 molecule of ethanolamine. And its molecular weight is 592. It's a pre-charged molecule, so that's very advantageous. It should fly really, really well in a mass spectrometer. This is the most well-characterized component of lipofuscin and is believed to be toxic to the retinal pigment epithelial cells. So it is a target of a lot of investigation in vision research in relation to AMD. So the question is, where is it in the retina? So the first thing we did was we looked at a mouse model of AMD. And so in the control mouse, we see in that particular flat mount preparation, sublimating dihydroxyacetophenone as the matrix. We see a signal at 592, shown in red here, and lots of lipids. So we get good signal, but when we knock out a transporter for the retinoids, the ABCA4 knockout mouse, we see that this 592 signal is the base peak in the mass spectrum. So if you want to image a signal, that's the one you really would like to image. It's so abundant. So as predicted, this molecule flies really well. So we have a standard for this molecule. We did MS-MS directly out of the tissue and on the standard, so we know that this is indeed the A2E signal that we are imaging. So in the mouse model, what we see in the control mouse, this SV129 mouse, at 2 months of age, we see a central distribution of the A2E signal. This is all 592, master charge 592 images. And then here in an older control mouse, we see an increase in the signal for 592. But when we knock out this transporter, at 2 months of age compared to the control, there's an increase in this retinoid signal. And then in the older knockout animal, the retina is loaded with this A2E molecule. So as expected, we get an increase in this retinoid metabolite. Now, unfortunately, mice don't have macula. So it's not the ideal model for AMD. So we looked in humans, and what do we see? Well, what we see is we see A2E present in the human retina. This is a 41-year-old human retina. And as we look in older retina, we see an increase in the A2E signal. But what is most important about these images is that the A2E is present in the periphery of the retina. The macula is in the center of the retina. It's responsible for our high acuity vision. So in no case did we ever see A2E present in the very center or macular region of a human retina. And this was also validated by LCMS experiments where we dissected out regions across the retina and quantitated A2E. So A2E itself is not present in the macular region, but these are normal human retina as far as we know. So what about in disease? Maybe it's abnormally abundant in the disease state. And it turns out that that's not the case. So this is a normal retina image. The dotted circle indicates the macular region. And you can see that there's no A2E present in the normal. There's no accumulation of A2E in a wet AMD patient or in a dry AMD patient. Yet the fluorescence, which is indicative of the lipofuscin accumulation, is highly abundant in the center of the retina. The dark spot is the optic nerve head. And these fluorescence micrographs show that in our preparation we see the RPE layer present. So here it's a continuous layer of RPE cells, and then you can see that these do not look normal as shown in panel C. So these are the diseased macular regions shown in a micrograph. So our conclusion is that the A2E molecule itself is not involved in the etiology of AMD. Now, it could be that other retinoid metabolites are involved. So our next step is to figure out what molecules are accumulating in the disease region using whatever sources we have available to us, imaging, of course, but even LCMS to try to dissect out what's happening in the macular region. So the last part of my talk, I'd like to talk about one of the bottlenecks and where I think at least one future area of investigation it will help move the imaging process and the imaging studies forward. So we're getting better and better at imaging specific analytes from the tissue. But the first question that the biologists ask is, okay, you got a signal, what is it? And that takes a lot more time to figure out what it is. And so for lipids, we can do, as Tim discussed, we can measure mass with high mass accuracy, the lipids from the tissue. We can do tandem mass spectrometry. But for intact proteins, that's a little bit more difficult. So in my laboratory, we've been taking a slightly different approach. We do a micro extraction using about 1.5 microliters of 20% acetonitrile. We apply this directly to the tissue with a gel-loading pipette tip. This samples roughly 2 square millimeters of tissue. It's not high spatial resolution, but we can still define regions of the tissue where we're extracting the proteins. We then take this solution, this extract, and bomb load it directly onto a C4 LC column or a C8 HPLC capillary column, and gradient elute the proteins into a tandem mass spectrometer where we use electron transfer dissociation to fragment the intact proteins. So we're able to get really beautiful fragmentation. And so the first point I want to make from this slide is we get beautiful electrospray mass spectra from this very, very small volume of extraction. So here is an electrospray mass spectrum of a signal at 22.6 kilodaltons from a bovine lens. This signal was imaged. So here's the tissue section that was imaged. That's the 22.6 kilodalton signal present. And then the ETD mass spectrum gives you C and Z ions, and all of this was acquired from that 1.5 microliter extract. This spectrum can then be manually interpreted or using ProCyte PTM, or Mascot in some cases helps interpret these spectra. This turns out to be beta A3 crystalline, and it's a truncation product where the N-terminus 22 amino acids are truncated. Here's another example, again from Lens, of a 20.7 kilodalton signal. Again, beautiful electrospray mass spectra from the extract, beautiful ETD spectra, and we can interpret this as gamma D crystalline. We've applied this also to brain tissue and to kidney tissue, and it works in a variety of different tissues. So now we can go in and relatively quickly extract the protein, get intact protein tandem mass spectrometry, and identify the signals. And those are the data that I'm anxious to look at on the cataract peptides I showed you at the beginning of the talk. So in conclusion, I think imaging in particular has important applications in ocular tissues. I talked about protein and lipid imaging and some metabolite imaging. We're also interested in drug distributions in ocular tissues. So I think you'll see a lot of applications in these tissues in the coming months and years. Spatial resolution is critically important, especially as we look in the rodent models. These tissues are very, very small, and so as we are constantly trying to improve our spatial resolution, I showed you 10 micron spatial resolution. We now have some 5 micron images, and so we're constantly pushing that, that frontier. And then lastly, it's important, very, very important to identify the analytes, as Tim pointed out. Otherwise, we don't know anything about the biochemistry and the biology that we're studying. So this remains a bottleneck. It takes time, but I think we're making some headway. And lastly, I want to thank the postdocs and graduate students and collaborators who've helped on these imaging projects. In particular, Gus Gray and David Anderson, 2 postdocs in my lab who've generated all the images that I showed you. And our funding sources from NIH and the support of the Mass Spectrometry Research Center at Vanderbilt. So thank you for your attention. I'd be happy to answer any questions.